Hello everyone, this is our second lesson in chapter number 10. It's about genetic recombination and gene linkage. So it's a very short lesson. Hopefully we're going to finish it pretty quickly. So what the genetic recombination is. So as you can see here, the definition is a new combination of genes produced by crossing over, and independent assortment is called a genetic recombination. So basically, once again, we have on this picture homologous chromosomes. So it's basically a pair of chromosomes, one from your father and one is from your mother. So basically, the crossing over happens. Try to remember, we studied while doing meiosis 1. So in prophase 1, crossing over happens. So on the third pair, on the third picture or pair of chromosomes, you can see that they exchange genetic material. So number of those combinations can be calculated by formula 2 to the power of n, where n is the number of chromosome pairs. So basically, as you know, the humans have 23 pairs of chromosomes, so it's going to be 2 to the power of 23, or we mentioned, for example, pea plants. They have less chromosomes, 7. There are some animals, plants that have more chromosomes, but basically that's the same formula. Very important for you to remember is that genetic recombination increases genetic variation. Now, try to remember the picture from a previous lesson. The picture about the chromosome and a gene that I pointed out. So let us say it was the gene about the eye color. If next to that one I draw another gene, for example for hair color or skin color, we can say that those genes that are located close to each other on the same chromosome are linked, and they usually travel together during the gamete formation. There are also some research on fruit fly, Drosophila melanogaster, that's a Latin name for that fruit fly, revealed that linked genes do not always travel together during meiosis. So as you saw, like in the third statement here, the gene linkage results in an exception to Mendel's law of independent assortment, because the linking genes usually do not segregate independently. Once again, there is a question, try to remember what the gamete is, try to remember the previous lesson about the genes, how they're located on the chromosome. Now try to imagine a chromosome with different genes, or you can use this picture here shown. It's showing the one chromosome of Drosophila melanogaster, or a fruit fly, with the different genes. So color of the body, then eye color, shape of the wings. Basically, those genes that are closer to each other on the map, they show a low frequency of crossing over. Or genes that are close together have a lower frequency of crossing over. Those who are further apart, crossing over occurs more frequently. Next term that we're going to explain is polyploidy. You already met the terms diploid and haploid number of chromosomes. So diploid refers to the 2n number of chromosomes, or 2 sets, and n, or haploid, refers to the 1 set of chromosomes. Polyploidy means that there are more extra sets of all chromosomes in an organism. Please do not connect this term with non-disjunction, which refers to cell division during which sister chromatid fail to separate properly, and it will occur the extra chromosome, not the whole set of chromosomes. So here we have a triploid organism that have 3n number of chromosomes, or another extra set of chromosomes. Here on the left slide, you can see some common plants. Usually the plants have extra sets of chromosomes. So triploid, like watermelon, banana, or blackberries that have 4 sets of chromosomes, or 8, even 8 you have strawberry. So that's it. With this one, we're finished with the first set of videos. Have a nice weekend.